Herkese merhaba. Bunu Türkçe değil de İngilizce yapmam herhalde daha doğru olur, değil mi? So, if I should say a few things about myself, I currently chair the Department of Molecular Biology and Genetics at Bilkent University, and I happen to be a medical doctor, or I should say I graduated from medical school, Ankara University Medical School in 1988. Then I pursued a PhD at Cornell University, Immunology and Molecular Biology. Then I remained a postdoc at the Memorial Sloan Kettering Cancer Center in New York as well for about 6 years. And then after that, I became an assistant member at the Ludwig Institute for Cancer Research within the Memorial Sloan Kettering Cancer Center for again about 6 years. And after that time, I joined Bilkent University in 2006. And in the last 14 years, I've been working there. So during the last 14 years, I became interested in transcriptomic research. Before that, we were doing translational research, cancer research, when I was in the United States. But it was not active transcriptomic translational cancer research. So translational research, as you know, means that we can communicate basic science to the clinicians and can communicate or understand clinical issues and try to generate an answer to them using basic science approaches. So I'm going to, we do this primarily using transcriptomics as an approach. You can do many types of translational research, and ours is related to transcriptomics. We also do non-cancer research. We currently are working with an in vitro fertilization clinic, trying to discover biomarkers that would predict the window of implantation. And we are also working with two different groups who are independently interested in bipolar disease and schizophrenia, and we are interested in transcriptomic biomarkers that could help clinicians understand the disease better and identify individuals with high risk. But today, I'm going to share with you primarily the research we have been doing in the last several years related to cancer. So I'm sure all of you know what transcriptomics is, but I have a very quick brief to tell you what it is about. So in our cells, we have about 20,000 genes, and the reason why we have so many different genes is because they have different sequences, and these different sequences exist because they are supposed to make different proteins, and the reason why we have different proteins is because we have different functions in our body. So if you look at the expression rate of mRNA for these genes in different tissues, you will see differences. A certain gene might be expressed in skin, but not in colon, and vice versa. Some might be expressed together, and some might not be expressed at all in these tissues, but expressed maybe in others. If you compare cancer tissue to a normal tissue, you will also find differences. There could be upregulation, downregulation, altered expression, and many more things. And if you compare different tumors to each other, you will also see that certain transcripts are expressed at different ratios in different tumors. This gives us the opportunity to differentiate between these tumors, although they might, to the pathologist's eye or from a morphological viewpoint, be similar. From a transcriptomic viewpoint, you might be able to identify differences between them, which you could not normally identify using pathological approaches. So transcriptomics is possible because we have high throughput methods that can generate transcriptomic data. Microarray technology and RNA sequencing are the two primary technologies that currently exist. What do you do with this technology? This technology gives you, for a given tissue, for example, for tumor 1, the expression rates of all 20,000 genes. An expression rate simply means a number. The higher the number, the more powerful the expression is. And therefore, we can say, we can compare different tumors. We can compare different tumors and identify the differences of gene expression in them. Then, if you have a clinical question, let's say you want to find out which of these genes can be a marker, and because it's a gene, which of these can be a biomarker for, let's say, overall survival. Then you do a very simple thing. You look at the gene expression rate and you look at overall survival. And if there's a correlation between them, you have found your biomarker. Although this looks very simple, the statistics you use to identify the relation between this gene and overall survival in this case are highly variable. You can be very creative here. You can use all types of tests, different approaches, and this is where, as I said, the creativity comes into play. And therefore, if two people look at the same data, they might end up selecting very different genes based on their approaches. But this is the primary concept. This has been used already. For example, in colon cancer, you can buy these tests which the oncologists use, and these list some transcriptomic tests like Oncotype DX colon, for example, which is a test which I believe costs more than $4,000, and it's used to prognosticate stage 2-3 colon cancer patients to see, therefore, how their survival will be. It's a RT-PCR test based on a 12-gene signature, but the discovery of these genes was through microarray technology. There are some other tests which use microarray technology itself as the test in the clinic. Similar to colon cancer, in breast cancer, there are many tests that with different outcome ideas. For example, some are meant to identify molecular subtypes. Some are meant to identify a certain risk score and so forth. So why should you do transcriptomics? There's a major alternative to transcriptomics, and that's genomics. So DNA mutation analysis based clinical categorization of samples. This works sometimes very well. For example, in AML, you can cluster samples based on whether they have a certain mutation or a different one. These mutations, as you can see here, are all mutually exclusive, so they can classify patients easily into subcategories, and these subcategories of patients have very different overall survival rates. So therefore, if a patient has P53 mutations, we know that the survival will be very bad, whereas RUNX1 and 1XT1 mutation individuals will have a relatively good prognosis. However, this is not true for most other cancers. For example, in gastric cancer, this shows a variety of mutations that might be driver mutations for gastric cancer and individual samples here. The samples indicated with red arrows are those for which no mutation has been identified. And as you can see, the mutation profile here is not easily classifiable. So for most tumors, especially solid tumors, a categorization based on mutation profiling is very difficult to perform. There are several reasons for this. For example, this is gastric cancer, and we are looking at the mutation profile of the 12th part of the primary tissue, 13th part of the primary tissue, 11th part of the primary tissue, lymph node 3, lymph node 1. These mutations are common to all, but these mutations are identified only in the primary tumor, not in the lymph nodes metastasis. These are found only in the lymph node metastasis. And here you can see individual mutations which occur only in a given site and which are not shared with the rest of the tumor. And similar pictures emerge if you do the same analysis for different tumors. So tumor heterogeneity makes it very difficult to classify tumors just based on their mutational profile. A mutational profile can also change very rapidly. This is a circulating tumor cell mutational profile in lung cancer. And you can see the variety of mutations here in a tissue that was obtained in March 2016. Only about 3.5 months later, the mutational profile changed dramatically and some of the mutations were completely lost. And it's possible that some new ones emerged. So therefore, the cancer evolves over time, and this makes it also difficult to recommend something to the patient based on just their mutational profile. So therefore, we are doing transcriptomics, and I'm going to show you that we are relatively successful in doing this. We performed two different types of projects in the laboratory, a top-down approach, that's how we call them, and a bottom-up. A top-down means that we have a clinical question in mind, let's say prognostication of cancer, and we don't know which genes are going to do this. So we first try to find out if there is a particular genotypic difference that could be identified using transcriptomics in these patients. Then we tried to compare the different groups that have emerged in this transcriptomic analysis to see if they behave differently, regardless what test you are interested in. Alternatively, if we start from a given gene, which is already known to be upregulated or downregulated in a given tumor group or a cohort, or let's say we have drug-sensitive and resistant groups, and then we try to identify the genes that correlate with this phenotypic, again, a phenotypic difference, then we call this a bottom-up approach. Since we are a translational research laboratory, we have these indicators, the technology and readiness level indicators, and you can also call them biomarker readiness level indicators. These mean that we are not strictly a research laboratory. We are trying to generate products that could be used in the clinic, and therefore we need the university setting for the discovery, but we also need an SME, which we do have. So two of my ex-students have set up their own small enterprise, and they are doing this type of projects. And obviously the reason for doing this is to bring the product to big pharma, who could then hopefully commercialize these. So I'm going to speak to you about our projects which fit into these categories, cancer-related projects and those which have been developed over several technology readiness levels. So I'm going to speak to you about our colon cancer subtype discovery and how this relates to different therapy options for these different subtypes. And this is also related to colon cancer prognosis. We similarly did something for gastric cancer. We have identified prognostically distinct groups. We attempted to do the same for triple-negative breast cancer and hematological cancer and pancreatic cancer. And recently, there's also data here from prostate cancer and melanoma, but which I'm not going to speak about today. This means that if we can get if we can get hold of samples, clinical samples, then we can do research in almost all types of cancer, and as you are going to see, hopefully successfully. Recently, we are also interested in RNA modifications. I'm going to speak about this briefly. There are many RNA modifications, and some of them are clinically relevant, and they can therefore be biomarkers of disease. So these are top-down approaches. For bottom-up approaches, we collaborated with an Italian group who had developed a SRC inhibitor, and they wanted us to identify which cells would be sensitive to the SRC inhibitors and which were the resistant, and generate biomarkers that could identify these different cells. We also collaborated with a group in Portugal, and they had discovered a particular gene which they thought was relevant in multiple cancers. And they wanted us to see if this related to prognosis in different cancers, and that's what we did as well. And I'm going to briefly speak about all of these to you today. So how do we do these analyses? We initially start with an in silico, strictly in silico analysis. Starting strictly with an in silico analysis means to download transcriptomic data from Geo or ArrayExpress. So I did this just 2 days ago. And there are, as you can see, 15 million or transcriptomic data from 15 million cancer samples in Geo. these 15 million samples come from clinical samples and also cell lines that have been treated or untreated, etc. But you can imagine the huge variety of samples here, which will surpass any cohort that we can collect on our own hands. So this is an extremely valuable site, and almost all of these clinical samples, if there are clinical samples in them, and there are, and the majority are clinical samples, all clinical samples have some clinical data associated with them. So by doing a transcriptomic analysis on the clinical samples and correlating the gene expression levels with a given clinical parameter, you can generate gene lists that identify that can predict that phenotype or that clinical parameter, or you can identify individual genes. So gastric cancer, I mentioned to you or showed you a few slides ago, is very heterogeneous mutationally, but transcriptomically, 20 genes will classify all gastric cancer into two clear groups. Depending on whether they are upregulated in red or downregulated in green, you can clearly see two groups. This is one cohort. It's a very large cohort in silico analysis, 300 patients, and here you have another one, 192 patients, and you can see two clear differences. So these genes were identified based on the fact that they should correlate with overall survival. And therefore, if you do a Kaplan-Meier analysis with samples that have high expression of these genes versus low expression, you can clearly see a very dramatic difference in survival. The patients with high expression of these genes have a median overall survival of more than 100 months, whereas the ones with high expression, I'm sorry, I think I made a mistake here. These are patients with low, and these are patients with high expression. And patients with high expression have a median overall survival of 26.2 months. So there's a very dramatic difference. In this cohort, we see a similar picture. Again, the patients with high expression of these genes have a median survival of 20 months, as opposed to those with low expression, which survive 50% or more of them survive more than 100 months. This is very dramatic, and it indicates a clear difference between these groups. So with gastric cancer, we have not progressed much, but this is as much as we did. We compared these groups with stage and multivariate analysis, and we find that this classification is independent of stage in both cohorts, and it is independent of the gastric cancer subtype, intestinal versus diffuse, which is a prognostic parameter in gastric cancer. Now we are pursuing the biology of this in gastric cancer, and we just obtained our samples with which we can validate these genes in our laboratory, and this is supported by currently a TÜBİTAK grant on gastric cancer. This is colon cancer, and this is based on just the expression of two genes which can prognosticate colon cancer. On the left, you see microsatellite instable, on the right, microsatellite stable tumors. This is an in silico analysis, and it shows recurrence-free survival time. We can predict three groups with good, intermediate, and bad prognosis based on the expression of just two genes, ULBP and SEMA5A. We published this data. This was supported by a different TÜBİTAK grant, and this was the most complete prognostication effort of ours. These genes were supposed to serve as an alternative to ColoDX colon, to that test which costs $4,000. We did a multivariate analysis using our genes and Oncotype DX. And we found that in the multivariate analysis that our genes could prognosticate colon cancer independent of age, gender, stage, and microsatellite instability status. And they did this maybe to an extent that was even better than what Oncotype DX colon could do. This is very successful in our opinion. And this we brought to a company, a pharmaceutical company in Turkey, and we patented this discovery, and we published this 3 years ago. So this is an example. These two were examples to colon cancer and gastric cancer. But in colon cancer, we went a little bit further. We said, if we can prognosticate groups like this, would there be drugs that we could recommend that patients with especially bad prognosis be given? Because we don't want to leave these patients as such, but we would like to give them a therapy option. For this purpose, we routinely use two large cell line databases. One is the CCLE, Cancer Cell Line Encyclopedia, and the other is the CGP, Cancer Genome Project. Currently, the Cancer Cell Line Encyclopedia has 504 cell lines and 24 drugs. Recently, they increased this to include many more. And the Cancer Genome Project had a few months ago 988 cell lines and drug sensitivity data to 453. Now, this is very important because if you have, for example, 58 colorectal cancer cell lines here, and you can model these cell lines to look like your tumor data, then you can ask whether the cell lines similar to the good and cell lines similar to the bad prognosis differ in sensitivity to a certain drug. And we know that the colorectal cancer cell lines transcriptomic profile is very similar to primary tumors. So therefore, our predictions for colorectal cancer will probably match the clinical samples. And we recalculate, we precisely calculate our IC50 values ourselves using in-house scripts. And we not only use IC50, but we use activity area, Amax, EC50, etc. parameters as well. When we correlate the activity area, when we correlate these 6 different models in CCLE and CGP data for the cell lines which have the same drug sensitivity levels, then we see that this model here works the best. This is the 3.3 PT 100 model, and it gives the most consistent results between activity area and IC50, and therefore we mostly used this model. I'm not going into details here, but I'm just trying to give you the impression that the downloaded IC50 data from CGP and CCLE need to be re-evaluated for proper use. Then we found out this. This is a drug, it's a PI3 kinase inhibitor developed by Novartis. And we found that this drug is more effective on cell lines that look like samples with bad prognosis in colon cancer, not as effective on samples with good prognosis. So therefore, at the end of the study, we could suggest BASE 235 from Novartis as a treatment option for patients with bad prognosis. This drug was undergoing trials until recently when it was stopped due to toxicity effects, but there are many more alternative PI3 kinase inhibitors that are being developed, and we look forward to seeing whether there will be a difference in treatment response to this drug in patients which are classified based on our gene, with 2 genes. We can identify or try to analyze the biology behind this prognostic difference in tumors. For example, this is on the top, a proteomic analysis in silico. We are comparing colon cancer with bad prognosis according to our subgrouping, good prognosis, and we find that EGFR tyrosine 1068 phosphorylation rates are lower in the bad prognosis group, higher in the good prognosis group. Caspase-7 cleavage products are higher in the bad prognosis, lower in the good prognosis. Now, EGFR phosphorylation happens to be a primary marker of epithelial cells and is less in mesenchymal. Caspase-7 cleavage is related to NF-kappa B activation which is related to transcriptional upregulation of inflammatory genes like IL-6, IL-1, beta, TGF-beta, etc. And indeed, when we do a transcriptomic analysis to see if these inflammatory cytokines are upregulated in bad prognosis subgroup compared to good, indeed, that's what we see. Since that's the case, we compared the EMT status based on E-cadherin expression and vimentin expression in colon cancer. And here you can see that tumors that have bad prognosis have generally high vimentin and low E-cadherin. And this is exactly a mesenchymal phenotype. So mesenchymal tumors have a generally bad prognosis. Epithelial tumors generally have a better prognosis. So this made the story kind of complete because we realized that the tumors with bad prognosis are actually mesenchymal tumors, and it seems the PI3 kinase inhibitor might work better on tumors with a worse prognosis. Now, this is a different study, similar in approach in triple-negative breast cancer, where we were trying to identify subtypes of triple-negative breast cancer that could be treated with different drugs. For this purpose, we separated triple-negative breast cancer cell lines in silico into two groups. Epithelial, which had not stem cell-like features, and mesenchymal cells, which had stem cell-like features. Then we tested using CCL and CGP whether there would be drugs that would be effective on these two groups differentially. We found two groups. Lapatinib is more effective on epithelial cells, and midostaurin, a multikinase inhibitor, is more effective on mesenchymal cells. So this is important in our opinion because they could be combined in triple-negative breast cancer, and if the tumor is composed of a mixture of these two cell types, these two drugs could be effective. Then we asked whether an epithelial breast cancer cell, in this case MD57, I think, could be switched to midostaurin sensitive phenotype by growing the cell 3D, meaning converting it into a more mesenchymal state. Indeed, that's what happens. If you grow the cell line in 3D, it becomes sensitive to midostaurin and, as expected, becomes resistant to lapatinib. Is this indeed EMT? If that is so, then the phenotype should be reversed using siRNA against ZEB1 or SNI2. And indeed, that's what we see. For example, here you can see that if you treat cells with shRNA to ZEB1, then midostaurin sensitivity is increased, and the opposite, and lapatinib sensitivity, if you treat the cells with ZEB1, then the cells become less sensitive to midostaurin, but they are going to become more sensitive to lapatinib. And if you treat so on the top we have midostaurin, on the bottom we have lapatinib, and here you see the differences. So this means that they are becoming more resistant to midostaurin. This means they are becoming more sensitive to lapatinib as expected. And we could see more or less the same thing for SNI2, but not in lapatinib case. Nevertheless, there was a consistent, more or less consistent profile here indicating that this could indeed be EMT and that these could be EMT-related drugs. If we look at prognostic subtypes in breast cancer, this is a complex picture, but it has been published, and this grant is still ongoing. It's again from TÜBİTAK. It's a Poland-Turkey collaboration study. We do not see consistently different prognostic subgroups in breast cancer. In other words, our success rate in identifying prognostic subgroups in gastric cancer and colon cancer did not reproduce itself in breast cancer using classical approaches. But recently, we have two different approaches which we applied, starting from a metabolomic approach, in silico metabolomic approach, followed by a transcriptomic approach, and that seems to be working, but the data I'm not going to show you here. However, this is what I wanted to show you. A graduate student in my laboratory recognized that in triple-negative breast cancer, there were two big groups that could be identified based on immune-related genes. There was a big group of samples that contained high expression of immune-related genes and those which did not. And these two groups indeed have different survival and in different cohorts. This is, for example, a Metabric cohort. It's a very, very large cohort and contains patients both from Canada and from Great Britain. And in both cases, we can see that patients with low immune gene expression have worse prognosis compared to those with high immune infiltration. And obviously, if you do a gene set enrichment experiment, you can see that in the group with high immune genes, the immune system processes are prevalent. But in the others, you have microtubule skeleton activity. So this is interesting because a drug that is used in breast cancer treatment, paclitaxel, is a microtubule inhibitor. Therefore, we asked whether in a paclitaxel cohort, the survival difference of these two groups would be minimized. In these, that's what happens. If you treat the patients with paclitaxel, the bad cohort, which has high microtubule activity, survives as good as the immune profile cells. But the overall survival rate of the immune group also decreases because paclitaxel is also an inhibitor of B cells, autoreactive T cells, and inflammatory cells, and therefore it's used in autoimmune disorders. So this clearly says that if you have triple-negative breast cancer patients, you have to distinguish them based on their immune profile first. If they are immune high, you do not give them paclitaxel, but if they are immune low, you give them paclitaxel. So this is something which we have not yet published, but it is going in that direction. We also tried to reclassify hematological cancer based on a transcriptomic approach, and this was a wild, risky project generated by a very bright graduate student in the laboratory. He generated transcriptomic signatures that could reclassify hematological cancer, and he claims that something called group 1, which could be identified transcriptomically, includes mostly AML samples, but some KML samples as well. Whereas group 2 includes mostly KML samples and maybe some AML samples. And these classifications are not nonsense classifications, they are real classifications, because if you compare the drug sensitivity profile of the classes which we have generated, you can see that for some of these, you get better treatment responses, like in the case of sorafenib, for example, group 1 tumors respond to sorafenib much better than any other groups. But if you use a classical clinical categorization, both AML and KML have a lesser response rate to sorafenib than G1. So the red squares here all show you the drugs which we see are more beneficial to patients if they were to be classified using our groups. So this is a relevant analysis. And it's an example to what we have done in hematological cancer. For hematological cancer, we had a second project, and this was a cost project led by an ex-graduate postdoc of mine who is now working independently. He generated a model based on several cell lines shown here in black, and this model could predict cell lines which were not part of the model as shown here in terms of sensitivity. And this worked very well for doxorubicin. It didn't in AML. It didn't work very well for other drugs, but for etoposide with some exceptions, the model still seems to be working fine. I also wanted to mention to you what we are doing about RNA modifications. So currently, Modomics, which is a database for RNA modifications, indicates that there are 172 different modifications that can happen to RNA. Recently, we were snobbish enough to call these decorations because we didn't know what their function is and was, and we still don't know the function of the majority of these. but it is impossible that they have no function. And we chose one of these, the A to I modification of adenine. So here you have the adenosine base, and here you have the deaminated version, which is therefore an inosine. So the A to I conversion is relatively easy to catch in RNA sequencing reactions because these A's become G's because inosine pairs with cytosine in reverse transcription, and ultimately this base is thought to be a G with RNA sequencing experiments, and therefore an A to G conversion is indicative of the fact that the I had been originally converted to inosine. Now, the A to I modifications are known to have clinical correlations. This paper, published 5 years ago, shows that these non-synonymous mutation causing A to I modifications have a relationship to subtype, stage, survival, etc. in many different types of tumors. We preferred to concentrate on the 3' untranslated region of cell lines, and we tried to correlate the presence or absence of A to I modifications in these with drug sensitivity, because this had not been done before. So this is the preliminary work of a very bright another graduate student in the laboratory who downloaded RNA sequencing data from cell lines and correlated the A to I editing ratio to sensitivity of different drugs. And here you can see 3 drugs and 3 different regions that contain a particular A to I modification. And you can see that that particular modification, in this case when it is high, indicates a very high sensitivity to this drug, but when the modification is non-existent, the cells are relatively resistant, similar to the others. And this is maybe one other example that I would like to give. This is purely an in silico analysis where we did no experimental data, we did no wet lab experiments whatsoever, but we could publish this recently in PLOS ONE. And the reason we could publish this relatively easily was because the gene signature generated by, Demirkol, again, a recent postdoc who is now a junior faculty in Hacettepe University, because she generated a gene list that could prognosticate breast cancer in 6 different cohorts successfully. And this was independent of other prognostic factors, and it served better than multiple other gene lists, and therefore it was published immediately, even in the absence of validation studies with real tumors. So I want to give you, in our last few slides, 2 examples, 2 bottom-up studies. This is the SRC inhibitor 10A, and we have here different primary melanoma cells on which we tested this drug. The primary melanoma cells came as part of a collaboration effort which we had with Hadassah Medical Center in Jerusalem. And in Israel, there's a lot of melanoma because there are a lot of white-skinned people, and it's a very sunny country. So they sent us primary melanoma cell lines with associated microarray data, and we generated, after a lot of transcriptomic analysis, I should actually show you this, various subgroups of these tumors, and we identified those, in this case, which had higher sensitivity to 10A, which was the drug we were interested in. But we tried other drugs as well, and we obtained sensitivity profiles of these and published them 3 years ago. Recently, we reanalyzed this data from a systems biology approach, and we realized that the genes related to sensitivity to 10A, which is this one, were also related to valproic acid, a histone deacetylase inhibitor. And this suggested to us that the use of valproic acid could increase, possibly, sensitivity to this drug. And indeed, here you can see 2 cell lines, primary melanoma cell lines, treated with 20 micromolar of 10A, and this is the sensitivity. If you add valproic acid to this, you see a significant increase in sensitivity in both cases. And this is very important because valproic acid, as you know, has been used in epilepsy and is relatively safe. So this is a repurposing of this drug should we decide to use this SRC inhibitor in the treatment of melanoma. Just another approach for you to keep in mind. And this was a good collaboration. Another group in Portugal, they were working on TRP2 expression, and they asked us whether we could find a drug that could target cells with high and low TRP2 expression. This again turned out to be base 235, and when they used this drug in their experiments, they indeed saw that it was related to base 235 sensitivity. And we also did prognostic analysis for them based on TRP2, and we could see both in melanoma as well in colon cancer that the gene's expression was prognostic, independent of stage, age, and gender, and this was therefore also published 2 years ago as part of a collaborative study. So most of this work, all of this work is done by several students and ex-students of mine, Seçil and Kerem and Can Türk have graduated from the lab, and now they are working as independent scientists, junior faculty in 3 different universities. Barış and Wakas and Ferit Sadati are my PhD students. Murat is an ex-master's graduate, but he still collaborates with us. And Ferit Ahatlı, Marzana are also master's students. Özgür and Özlen are collaborators of ours at Bilkent University. And as I briefly mentioned to you, we collaborated with various individuals at different locations who provided us with actual samples. So thank you very much. If you would like to see a recording of the Turkish version of this presentation, you can go to my YouTube page if you search it with my name. And this is the title of that talk, Kanser Araştırmalarında Güncel Moleküler Yaklaşımlar. Here you are going to see me talking, as well as Seçil talking, as well as Barış talking. So I'm going to give a general overview of what we have done, and then you are going to see how a junior faculty who has graduated from our program is capable of explaining herself very nicely. And then you are going to see how one of my students sees his future in here. But I will also upload this video if you allow me to, because I recorded it on my web page, or I can give it to you and you can upload it if you like. So thank you very much. I'll take any questions in English or in Turkish as you like.
Thank you very much. I think we have a question from the chat. Maybe Mert Şahin can ask his question by using the microphone.
So while Mert is getting ready, I can rephrase his question. He wrote, isn't it a problem that different approaches producing different correlations between overall survival and gene expression rate? I guess you are trying to say, is it a problem that different approaches will generate different correlations between overall survival and gene expression rate? It's not a problem because the best correlation should hold true for most cohorts. It should be a very stable correlation. And we don't know which correlation is very stable unless we have extremely large cohorts. So therefore, it's good to generate different gene lists for different tumors that can predict whatever you like, overall survival, recurrence rate, sensitivity to drugs, or anything you like, and then test whether this holds true in different cohorts. You have to do a lot of validation studies, in other words, and validation can be a very, very long process. Therefore, as you remember, I showed you some transcriptomic tests that are out on the market today, and there are several of them. some people use one, the other use the other, and the more the validated the test is, the more applicable it becomes. I guess this is the answer to this question, probably. Would there be other questions?
We can switch to Turkish if there's anyone hesitating to ask English questions.
So before you ask questions or while you are thinking, I am considering close collaborations with the Acibadem group, both scientists and clinicians, and this talk was meant to serve as an introduction of my interests to you. If you are interested in working with me, then please contact me. You can find my email, telephone, and everything, and we can start thinking as to what we could be doing together should we be working more closely on the long run. And today, as your dean mentioned, I will be here, or actually as Uğur Hoca mentioned, I will be here another few hours this afternoon at Acibadem, should you want to see me.
I want to ask a question. Is it possible? When you use valproic acid against melanoma, did you measure the side effects also?
So this was strictly a cell line experiment. We just added valproic acid in vitro to cell line cultures together with the drug that we were interested in, the SRC inhibitor. So the side effects, the concept of a side effect is an in vivo concept, and we haven't done any in vivo experiments, and therefore I can't answer your question. But the side effect of valproic acid is very well known because this drug has been used in the clinic for many, many years. And we know exactly in what concentration it does what. So therefore, it is a good idea to combine a cancer drug with a known drug that has a different purpose, because we know more or less what its side effects are going to be. This is also called repurposing of a drug. So valproic acid currently isn't used as a cancer drug, but it could be combined with a SRC inhibitor. That's the take-home message of our paper.
Thank you.
Thank you. Mikrofonumuz kapalı.
Ali Hocam, sunumun gerçekten record'unu almak isteriz sizden. Biz de izniniz olursa kendi YouTube sayfamıza da koymak isteriz.
Tabii.
İsterseniz hani tartışma açısından